Okay, so we're talking about recombinant technology. That's when genes are removed and put into other cells. Um, it's a combination of DNA from two other cells, I guess. There's cloning with the somatic cell, nuclear transfer. Um, the DNA is removed from the female cell, put into an egg, and then the egg is put into a surrogate, and then it grows. What causes the cell to divide and grow are spindle proteins. Um, then there's DNA libraries. It's a sequence of proteins. So if you need to go back and look, so if you don't know the DNA, but you have the proteins, I think you can go back and look and put the DNA sequence together. Then there's polymerase chain reactions, so PCR. It looks like these are the copies uh, of a portion of DNA. They can be developed and grown in a cell or in a lab. There are three steps to that. There's denaturation. This is where the DNA molecules are separated. And then annealing, which is when the DNA is cooled and then they're put together with primers. Then step 3, which is the DNA synthesis, and that's when DNA is then developed and copies are made. Then we have gene therapy. This is interesting. The DNA, it can be missing, and the um, the errors can be fixed in the cell. The DNA can be replaced. The gene therapy is used for to prevent diseases, transferring genetic material from cells to proteins, can help reduce the level of proteins that are diseased, increases the good proteins in the cell. It can make new or change the proteins. There is in vivo and ex vivo. These are used to administer the gene therapy, so to help with it. So in vivo is when the gene is directly inserted into the person, and then ex vivo is when the cell is removed, and then the genes are then put into the cell and then put into the person. There's genetic engineering. That's when you can change an organism's genotype. Have an example. So if your pancreas isn't creating enough insulin, the insulin can be created in a lab and then you can consume it. So bacteria can be used to help produce insulin because bacteria divides pretty rapidly and creates generations pretty rapidly. So DNA is put into the bacteria and then it's multiplied and then you can consume that. Genotype and phenotype. So genotype is the pairing of alleles. So capital P and lowercase p, capital P, lowercase p, lowercase p. So the pairing of those. And then the phenotype is the expression. So what will be dominant and seen. Mendel's experimental design. So that's Gregor Mendel. He used pea plants to understand genotypes and phenotypes. He grew a bunch of the same pea plants, and then he started crossing the traits to see what the outcome would be. So he would cross two tall plants, and they might yield um, smaller plants. So then he discovered that the hidden genetics could then become dominant throughout the generations. Let's see. There are the three principles of inheritance, which is the principle of dominance. So that's how the genes that control phenotype actually work. There are always two copies of genes for a certain trait. So a capital letter and a lowercase letter, the capital letter and a capital letter. Dominant is always expressed. Then there is the principle of segregation. So that's how the alleles are passed on from the parent to the offspring. Each parent shares one allele that will be passed down. Then there's the principle of independent assortment. So every allele is split up from its partner independently. All the other alleles in the genetic code. I'm not entirely sure what that means, other than that creates variety. By saying that one plant can have dominant genes, dominant alleles that might not be seen in the offspring, then that offspring can have other offspring, and then those would show some dominant traits. Complex traits, from what I understand, this is there can be multiple dominant traits in one cell, and the traits can be affected by the environment or behavior, like behavioral factors. There's diploid organisms. So that is when there is two sets of chromosomes. There's haploid organisms, so that's one set of chromosomes. And there are the alleles, which is what is expressed. So what dominant allele is expressed. There's mono and dihybrid crosses. So you develop that with Punnett squares. So the monohybrid cross is when you have a single characteristic that you're crossing. And then the dihybrid cross is when you have two or more characteristics you're crossing. There's neo-Darwinian synthesis. I'm not sure I completely understand neo-Darwinian synthesis. It sounded like it was just talking about natural selection and evolution, which makes sense. But I'm not sure why it's neo-Darwinian. I don't understand that. Then there's how to statistically analyze genetic data using a chi-square test. So I did do that. I did the Punnett square, and then I made some, like, I think that's what this is, right? Chi-squared? I'm not sure. But I did this because I looked it up and I thought this was a pretty good idea. And that also goes along with the prepare an activity for the class to participate in statistics. So this is a statistical test here. This is the p-test, I believe. I think so. Like where the p-value, so it tells you if your findings are significant or not. This is pretty cool to learn about. Something else was positive and negative effects occurring in the population because of technologies. So, let's see, positive effects would be improvements in healthcare, like and diseases. So, um, we can, I think we have like the designer babies. I think we talked about, and we can change we can change those negative. Um, Genetics that we have, which is pretty cool. Maybe not ethical. I don't know. I don't know. I wouldn't do it, but I don't know. And we can also use that for crop diversity and like what crops we can make newer crops that maybe deter certain insects or things that are going to destroy the crops. Um, I think that that's pretty cool. Although I think that we have like plants naturally in the environment that do that, but maybe that's too expensive for us. So we go a really roundabout way of doing that. I don't know. Maybe we try to find more plants that we can use to deter bugs and other um, harmful creatures that take out crops. Then there's the negative modifications. So there can be side effects when crossing different plants or um, trying to develop a new um, health thing. Maybe that would be maybe that would cause negative side effects because something that you weren't expecting to happen could happen. So maybe going in and trying to mess with certain genes could actually cause negative genes to come about, or maybe it could cause a disability. Um, uh, Maybe it could even make health risks, like maybe changing one dominant trait would take away or affect another dominant trait that you might want. I don't know. But anyways, so what I did here, I'll explain what I did here. I hope that you can see it. So what I did was I just came up with numbers. Um just random numbers. Like I have my phenotype here, which is purple flowers and pink flowers. And then I have my observed plants. So I think it'd be really fun to have like a crop of beans or something or plants or something. I don't know. I think that'd be really fun, but I don't think we have time for that. So I just pretended. And I said that I have 6,256 purple flowers and 2,301 pink flowers. And then I added those up. So then I would have planted and observed 8,557 plants with purple and pink flowers. And let's say that I expected to see 6,417 purple flowers, and I expected to see 2,139 pink flowers. How I got that was I did the Punnett square, and I just did capital P is my purple flowers, and lowercase p is my pink flowers, and I just crossed them. This is a super simple one. I didn't want to make it too complicated, but this is a super simple one, and I came up with 75% of my flowers total are actually purple flowers, and only a quarter of them, so 25% is pink. So I had dominantly purple flowers. And then what I did was I subtracted my observed flowers from my expected flowers. Okay. And I got 161.75. And then I did the same thing with my pink flowers and came up with 161.75. But I've got a negative and a positive up here. And then I squared those. And then I divided them by the expected ones, by the expected amount here. Okay. And that gave me 16.307. And that's really important because that means that I have no significant difference. Like if this was valid and real, there wouldn't be any significant difference um, between my observed flowers and my expected, like what came out of that. Um, but I thought that this was fun. This was kind of a way to see statistics. Statistics are kind of important. So, and then I just explained the p-value chart up here and how um, your p-value needs to be less than 0.05, and mine was at 16. So, when it's less than, it is significant, which is important, and when it's above that, it's not significant. But anyways, that's what I did. As for the activity, I don't know how to really prepare that. But I thought that this was fun. Maybe what there could be dice, using dice and rolling dice, and what you would consider what numbers, but then that would be extensive because there are typically uh, let's see, yeah, 6 sides on a dice. So then you would have 6 different numbers and 6 different outcomes. Or so that seemed a bit complicated. But maybe if you just used dice, that might be fun too. Maybe you could put stickers on the dice instead of numbers. That would be even more fun. Okay, thank you.